Hello, and welcome back to this scientific ethics lecture on DNA analysis. And so what we're going to be doing today is continuing our view of studying the genetics and how we can use the genetics to potentially understand human disease, as well as other diseases as well, but then potentially learn how that we can treat the disease. And so what we're going to look at today initially is the process of genomic sequencing. And so this is a little bit different than what we talked about in the previous lecture, where we took a look at like refraction fragment length polymorphisms, that really complicated kind of sequence of terms that basically says that we can kind of chop up our DNA into kind of unique and distinct patterns and use that to identify individuals. And we could use that to identify paternity, who is the father of a child of unknown paternity. We can take a look at taking a look at forensic DNA evidence, as well as the 23andMe or the Ancestry, those DNA kits that you kind of put your saliva in a tube, you send it off, and it comes back and tells you a lot about who you're related to and the heritage. What we're going to be looking at today is a little bit more kind of finite detail, a little bit more specific. Instead of looking at kind of big chunks of DNA that are cut up with those restriction enzymes, what we'll be looking at is the exact sequence of the A's, the T's, the G's, and the C's, those nucleotides that are carrying the code for the information that we're interested in. And so we're going to start out a little bit with the science and then talk in the second half about some implications on how we can use this, again, to study human disease. So just as a quick reminder, if we take a look at our DNA molecule, it looks kind of like this ladder, where we've got the A's, the T's, the G's, the A's, the T's, the G's, and the C's are those nucleotides forming like the rungs to the center of the ladder. And these are going to be the important things that are carrying the information. The kind of vertical sides of the ladder are just other portions of the DNA molecule that hold it together. Now, in the normal process of copying DNA, something the cell does when it divides, what's going to happen is that these two strands are going to be complementary to one another. That basically says that, if you've got an A on one strand, we're going to have a T on the other strand. If we've got a C on one strand, we've got a G on the other strand. In this process of replication, basically think about it as making a replica of our DNA molecule. We're going to open it up, and again, not worry about the enzymes and the chemical reactions, all that stuff. Just understand the basic process. We can open it up, and again, these vertical kind of portions of the ladder are still going to remain intact, where we're separating the A's to the T's, and we're going to separate the G's from the C's. So when we do that, what we're ending up with is one strand open over here, another strand open over here, and it's going to be complementary. So where we have an A, we can put in a T, in this kind of lighter blue. Where we've got a C, we can put in a G. And so we can fill in, based on this template, kind of the shapes, these A's, the T's, the G's, and C's on this one strand. We can put in these individual nucleotides, kind of line them up in that proper sequence so that we don't get a mutation. We don't get a change in our genetic information. On the other strand, in this case, we had an A. Over here, we've got a T. And again, the same complementary procedures occur. Where we have a T, we're going to put in an A. Where we have a G, we're going to put in a C. So we're going to bring in the individual nucleotides. We're going to line them up based on that pairing of the bases, the A's, the T's, the G's, the C's, like that. It's basically almost like a little jigsaw puzzle that we're going to put together dependent upon kind of the shapes that are present here. So what we end up with at the end of this process, we started out with a single DNA molecule looking like a single ladder over here. Again, A bound to T, C bound to G. If we take a look at over here, we got A, C, T, A, G. We're going to open it up, bring in the nucleotides, do some kind of magic chemical processes over here, kind of bind the vertical portions of the ladder together. So what we have going from one molecule over here, one DNA molecule over here, through the process of making a replication, into two molecules over here. And we can take a look at it. And they're lighter blue so that we can tell the difference of the new things we put in. But we've got an exact copy of what we have over here. So we get A, C, T, A, G, ACTAG, and on the other one here, even with the new nucleotides, ACTAG. And so we were able to build this molecule, keep the information intact because we've got this complementary sequence being present. And so this is important because this allows us then to make copies of the DNA. And this is something the cell does on its own without having to do that. But if we want to really study the DNA molecule and be able to understand what the sequence is and what the information is coded within the DNA, we're going to have to go through and understand that specific sequence. It's easy to do it in a cartoon over here, but in real life, it's a little bit more challenging. So what we're going to do in the laboratory is a process of artificial replication. We're going to replicate it in a way that we're going to do the same basic thing. We're going to start out with our DNA molecule over here. We're going to cause it to break apart. We're going to bring in the nucleotides, and we're going to fuse those kind of parts, those vertical parts of the ladder together. And we're going to do it, and then we're going to do it again, and we're going to do it again, and again, and again, and again, and again. And this is going to be a process of polymerase chain reaction. Again, a nice scientific phrase. Sounds really impressive when we take a look at it, but it basically, if we want to think about it, polymerase is going to be an enzyme that makes a polymer. And so our DNA molecule, when we take a look at it here, is a polymer because we got these individual little subunits that are hooked together into a long chain. So this just means that we're going to be making a long chain, and it's going to be this chain reaction where we're going to go from 1 to 2 to 4 to 8 to 16, 32, 64, 128, 256, and so on and so on and so on. Can't do that math in my head. So basically what we're going to do in the laboratory setting using polymerase chain reaction, often abbreviated as PCR for polymerase chain reaction, is this process normally occurring in the cell of replication. We're going to do it artificially within the test tube. And so we're going to end up with lots and lots of copies of that sequence of DNA that we're interested in. And this doesn't tell us the exact sequence, the A, T, G, C, order when we take a look at the nucleotides, but this gives us a tool in which we can start to maybe kind of understand the DNA. We can end up with lots of copies of the DNA that we can go in and study. Now, about 30 years ago now, was really exciting when it was occurring. It's kind of history now, but we started out with the Human Genome Project. It started out as a 15-year project, started in 1990. The goal was to go through and identify and sequence approximately 20 to 25,000 genes of the human DNA. And this was a joint effort that was done by the U.S. Department of Energy and the National Institutes of Health. So we see the NIH and National Institutes of Health popping up a lot in here. I'm a biologist. A lot of the examples I'm going to use are going to be biomedicine because there's a good relationship between the science and the ethical quandaries that I'd like you to think about as we go through that course. So basically, they went through and techniques that are commonplace today were experimental and being developed 30 years ago. But basically what happened is they discovered a way in which they could use that polymerase chain reaction in order to sequence the DNA, to find the specific, in this case, ACTAG, the specific sequence of those nucleotides that's coding for the message within the DNA molecule. And so in the Human Genome Project, what they wanted to do was to sequence the genes so we could better understand the DNA and potentially what was going to be occurring in the normal DNA so that we could then study the DNA of diseases and again, be able to understand the changes that occur. Again, many diseases have a genetic component, whether it's the genetics cause disease or the genetics cause a the genetics cause a predisposition so that there's a likelihood that you would get the disease. And so what they did is they basically went through and did polymerase chain reaction with, and again, I'm not going to go into the science behind it too much, but basically an ability to kind of end the DNA molecule at specific sites. And at those specific sites, we would know in this case, we added an A and it stopped. In this case, we added a C and it stopped. And so we're going to run the PCR, the polymerase chain reaction, make many, many copies of it, but we're going to put it in, so sometimes it stops at an A, so we'd have it stopping here and here and here and here and here and all the little green little spikes that we see here. Sometimes when we do it, we're going to stop it at the C's. So we got the C's and the blue kind of showing up here. And so if we do that gel electrophoresis, again, the example I gave, you know, the semi truck and the sports car kind of zooming down I-95, the small molecules are going to move through the gel relatively quickly. The larger molecules are going to take a little bit behind. We're going to do this in a way that we can separate these DNA fragments, these DNA molecules, not based on the big cuts with those enzymes that we talked about in the last class, but minor differences, a single nucleotide difference, kind of line it up. And this allows us then to sequence the DNA. And so with Human Genome Project, was thought to take 15 years. Once they got the technology developed, it went much, much faster than that. So they've been able to sequence 3 billion nucleotide base pairs of the human DNA. So it's like coming up with 3 billion individual letters in sequence on these DNA molecules on the chromosomes. And we're able to store that information in the database and start to use it. But the problem is, once we've got that information, what do we do with it? How do we use it? Because that, again, starts to enter into a series of ethical, legal, and social issues that the technology kind of introduced, because it's amazing technology. It's information that we've never had before. But now that we have it, how is it that we're actually going to be using it? Based on the success of the Human Genome Project, they basically went through and started sequencing other genomes. And so many species that are used in biomedical research, the bacteria E. coli, the Drosophila fruit fly, the laboratory mouse, they've done rats. Now at this point, they've done, you know, chimpanzees and gorillas and lots and lots and lots of different animal and plant species. Over 7,000 species have already been sequenced, and more being sequenced all the time. And they keep being added into these databases, which allows us then to, again, recognize the similarities and the differences between the genes of humans and the genes of different organisms. And if the genes of the different organisms are very similar, we can then study that gene or study that protein in a system without having to do clinical studies on human beings. But what this has done is given us huge, huge, huge databases full of genetic information. And so that caused a rise in a new field, which is basically kind of combining the area of biology with the area of computers, because we've got these huge databases, but then we got to try to figure out what it all means. And so in that field of bioinformatics, they go through and they start to program search algorithms to look for specific sequences. They do data mining to look at the similarities and differences of the DNA molecules. And they start to generate computer models. And these models then, again, are often going to tell us what's going on within the DNA, and gives us an idea what's going to be going on in the proteins that the DNA codes for, and what's going to be going on in the cells that are going to be expressing or using that DNA in that process. And so, you know, in a relatively short period of time, roughly 30 years, we've gone from not really knowing the sequence of the DNA to really being able to study the DNA across many, many different species and see the interrelatedness of that. Now, the databases that store the DNA information are queried and analyzed by scientists. So, you know, this is just a collection of 3 of them that are commonly used and are readily available. There's the Human Genome that's, again, all of these are on the NIH Gov's, the National Institute of Health government websites. And so you can go through and study the human genome on this website. You can study the relationship of different mammalian genes with another database. And then finally, GenBank is this huge collection of human DNA and animal DNA and plant DNA and bacteria DNA and all kinds of other different living organisms that we can now go in and study because we understand what that specific sequence is. We understand the code, and now we got to try to decipher it, figure out what it all means. Related to this half of the lecture, I've got a couple a little bit older articles now at this point, but it does describe the Human Genome Project and how the DNA sequencing process works. It's changed a little bit now with advanced technology, but this provides a good basic understanding of the approach. Even though the technology has changed, the approach is still the same. And so I'll post these on Canvas as well. So that's kind of how we can go through and study the exact sequence of the DNA molecule and be able to hopefully begin to understand it a little bit more. Come back for the second half of this lecture where we're going to start to talk about how it is we can start to use this information. As always, if you ever have any questions about this or other things in the course or other things that are concerning you, feel free to email me. Thank you and have a great day.